So here we are up in Hamilton, New York. It's a nice fall day, and we're looking for something very special. We're looking for an aquatic green algae in the genus Chara. And we have found it here in this ditch. You see this ditch is loaded with Chara. And the reason why Chara is so interesting, see all this stuff in here is Chara, is that Chara, this group in the Charophycine green algae, shares the most recent common ancestor with land plants. Your book refers to this as a green plant, fine, but this group, Chara, is the sister group that shares the most recent common ancestor with all land plants. It's fairly common in these roadside ditches here in Hamilton, upstate New York, and we're going to go take a look at some of the features of Chara to understand the positioning and the homology of the egg-bearing organs. Okay, so we're no longer out in the field, obviously, and I've brought you in to talk a little bit more about the genus Chara. Specifically, everything you see in this line drawing or this illustration is the gametophyte phase of the life cycle. Remember, the dominant phase of the life cycle in algae is the gametophyte phase. Specifically, I want to draw your attention right here to what this figure calls the reproductive organs. If you enlarge those, you actually see that there are two different gametangia. This large gametangium here is called the ogonium. That is the egg-bearing gametangium in the algae. This gametangium that subtends that, or right below that, is called the antheridium. The antheridium is the sperm-bearing gametangium. Now, the ogonium and the antheridium are actually homologous to all of the gametangia we see in land plants. And the green plants, as your book called them, they're a little bit different. The name changes in the algae from ogonium to archegonium in the land plants, but the antheridium, the term antheridium stays the same. The important thing is that one bears the egg and one bears the sperm. Now, if you further enlarge the ogonium, this is what you see. This is a scanning electron micrograph of the ogonium itself. And the first thing you notice is that there's a spiral cell here. This is a single-celled gametangium or ogonium. There's one single cell that spirals around the egg that is encased inside the archegonium or the ogonium here. Not much structure to this. Now what happens is the sperms are released from the antheridia, they swim around, they fertilize the ogonium. And when that happens, the zygote is formed. Now these plants undergo zygotic meiosis. So as the zygote begins to expand, it actually ruptures through the ogonium wall as it undergoes meiotic divisions to produce those 4 spores. Okay, so you can see even if Chara wanted to hold the zygote in, it couldn't do it because there's just not enough structure for that here. Now I have 2 other images here. First we'll look at this one. You see once again these images are not scanning electron microscope graphs, they are actually light microscope pictures. And again, you see in this one, you see the spiraling cell wrapping around that, and this darkened structure inside here is the actual egg. You can see the egg here. And if you were to sort of step out a little bit or zoom out a little bit, you would see that there are many of these ogonia on the body of the gametophyte of Chara, and just right below it are the antheridia, loaded with antheridia. So again, everything you see here is the haploid or the one-end gametophyte tissue in the genus Chara.